History of soil microbiology. The soil microbiology started to develop from last century. The possible existence of soil microorganisms in soil to have suggested by Columella in 60 BC. The decomposition of organic matter by soil microbes in soil in accumulation of nitrates and is therefore associated with soil fertility suggested by J.B. Bossing-Gold. The first who also showed that legumes can obtain nitrogen from air. Bacteria from root nodules of legumes was first isolated by M. W. Benzernik. Authentic drawings of microorganisms were made for first time by Antony von Leeuwenhoek. The discoveries of autotropism in bacteria and microbiological transformation of nitrogen and sulfur by S. N. Vinogradski. Cellulose degradation by anaerobic bacteria was discovered by Omlenskaya. Study on ammonification of organic nitrogenous substance by soil microorganisms was initiated by Lippmann and Brown. Study of rhizosphere was initiated by Hiltner. Importance of protozoa in controlling bacterial population and activity in soil was first suggested by Russell and Hutchinson. Context slide technique for study of soil microorganisms was initiated by Rossi and Colonti. Intensive study on mycorrhiza was initiated by Rayner and Malin. Penicillin was discovered by Alexander Fleming. Streptomycin was discovered by Waxman. The study of phylosphere was initiated by Ruinen. Essentiality of molybdenum for accelerating nitrogen fixation by legumes was demonstrated by Bortels. Essentiality of red pigment in legumes root nodules for nitrogen fixation was first proposed by Kubo. Isotope method to quantify the amount of nitrogen fixed was given by Boris and Wilson. The biochemistry of nitrogen fixation by root nodules in legumes was elaborated by Bergensen. The theory of microinvasion of root as explanation for origin of infection leads in root hairs of clover was proposed by Nutman, and also the heredity mechanisms behind nodulation in legumes was first elaborated by Nutman. The actinomycetes endophytes Frankia was first isolated by Chalan, Del Teredici and Torrey. Rhizobium from root nodules of non-legumes was first isolated by Try-Nick. Nitrogen fixation by Azobacter was first demonstrated by Debournier. The concept of associative symbiosis and diisotropic bioeconosis was proposed by Debournier. Nitrogen fixation by stem nodules of Succesbina rostata was discovered by Debournier. The soil microorganisms were classified as autogenous and gymogenous by Vinogradski. The technique of enriched culture was developed by Vinogradski and Benzernik. The technique of enriched culture was developed by Vinogradski and Benzernik. Direct microscopic count technique was developed by Kohn. The 15N technique for quantitative estimation of nitrogenous activity was developed by Boris and Wilson. Thank you everyone for watching.